Thank you very much and good evening. Welcome everybody to this evening's Bluetongue virus update webinar, part of an ongoing series of webinars that's been organised by the UK Ruminant Health and Welfare Group and facilitated by AHDB. My name is Colin Mason and I'm chairing the webinar this evening and I'm representing the British Cattle Veterinary Association on this occasion as chair. So just a few housekeeping points in terms of the webinar. The webinar is being recorded, and for anybody that has registered, it will be circulated within 24 hours if you want to catch up on any points within it. This is the third technical webinar providing an update on Bluetongue in England. There will be one on alternate Wednesdays, and please register for each one individually via AHDB. Our aim is to keep you updated with various different aspects of this disease as it progresses in coming months. We will hear from our speakers this evening, and then there will be an opportunity for some of your questions afterwards, depending on time. We've got a large number of attendees this evening, so please bear with us, and we'll try and answer all your questions as well as we can. Please, could you put your questions in the chat throughout, and then we'll have access to all of those and can choose them and try and answer as many of them as we can. Please also, we would really appreciate if you could complete the feedback form at the end of the webinar. It helps us plan future webinars with additional questions and topics that you're interested in relation to this subject area. So tonight's speakers and topics, we intend to start every one of these webinars with an update on the current situation in England. And tonight we're going to get a BTV update from Sasha van Helvoort, who is the veterinary head of field delivery at APHA. And then what we also felt we wanted to focus on this evening is some wider information about Schmallenberg virus as well as Bluetongue 3 virus, both vector-borne diseases, both affecting holdings in the south of England with similarities occurring as well. So we felt it was quite appropriate to look at what might be seen in lambs and calves that might be being born or being aborted at this time and how they relate to potential surveillance for Bluetongue and also what to consider about Schmallenberg as well. So we've got two speakers, Rachael Tarlinton and Vanessa Swinson, about that as well, and then we'll have a Q&A session at the end. Just a few other signposts as well. The Bluetongue Resource Hub is online through the Ruminant Health and Welfare Group website, and we update that as frequently as possible to give you the most pertinent updates on a whole range of information in connection to what's going on. Similarly, if there are concerns, there is a hotline and the number is there for you to phone to place a call so that we can try and answer any specific concerns that you might have. Also available on that website, there is an information leaflet about BTVA, which is BTV, which is available to download and to share, and is another useful resource of information. We would also like to signpost organisations where additional support is available. They are the logos of those are on the screen here, both to support farmers and vets through what we recognise can be quite a tough time in relation to this disease at the moment. So thank you very much just for listening to that brief introduction. What I'd now like to do is introduce our first speaker, who is Sasha van Helvoort. She is head of veterinary field delivery at APHA, and she is tasked tonight with giving us a quick update on the current situation in Kent and in Norfolk with Bluetongue, and an update on relevant facts from an APHA perspective. So Sasha, the floor is yours.
Just going to turn on my camera. Just hope you see what I'm seeing. Is my presentation up? Oh.
It's
not quite yet.
No, sorry. I'll try again.
It's currently just the browser machine, yeah.
I'll try it again.
if it's not playing ball, we could always we'll give it another second. But if maybe Rachel, if we could always move ahead in the agenda and come back to Sasha potentially.
Yeah,
But we'll give it a go now. But just to give Rachel the heads up potentially.
I'll
Give it
try
another
one more
second.
time. Yeah, I
There we
think
go.
this
Perfect.
is
There you go.
working. Is that full
Excellent.
screen now? Yes, brilliant. Right, sorry about that. A bit of a struggle bringing this on. Yeah, thanks, Colin, for the
Sorry, Sasha,
introduction.
you're in notes mode currently. If you want to click display settings at the top, and then
Oh,
it gives you the option to swap screens.
great.
Yeah, that should now fix the problem, hopefully. Swap
Yeah,
presenter view and Sasha. Yeah, there you go. Yeah.
is that it?
Yep.
Good.
Perfect.
Sorry about that. Great, all this technical stuff. Thanks a lot, Colin. And my name is Sasha Van Elvoort. I am Outbreak Veterinary Head for APHA, and continuing on with the update for Bluetongue with a slight recap, just for those people who haven't been on any previous webinars. So as you're all aware, we started with the zone near Kent and Canterbury in Kent, and it started with the 10-kilometre zone in the little circle there on the left side, and that was on the 11th of November, and that then got extended on the 4th of December. And then also we also had a further TCZ, a temporary control zone, put in on the 8th of December, and that was in Norfolk. It is called the Norfolk Temporary Control Zone, but there is a tiny bit of Sussex in that one as well. And then you can also see on the large map both the zones together. So as an update, as of the 23rd of January, so that was yesterday, but that hasn't changed today, so it's the same. There were 16 cases in Norfolk, which involved 16 cattle and no sheep. This is on 10 different premises with 9 different keepers, because some keepers have different premises, so we've kept that kind of separate. And of all those animals were humanely culled until the latest one, which is the one we had yesterday. In the Kent area, we've had 40 cases, 36 cattle and 4 sheep, and all animals have been humanely culled there on 24 premises and 14 different keepers. I note that I've actually done this the wrong way around, because one from yesterday was in Kent. In total, we've had 56 confirmed infected animals. There's 52 cattle, and that's been a wide variety of animals: dairy cows, beef, replacement heifers, finishing cattle. So it's different. And we've also had 4 sheep. And this has all been in 33 different premises with 23 different keepers. Obviously, we have the 2 temporary control zones in place. Of these infected animals, so these separate cases, there was no history of imported animals at all. We did have 1 case of maternal transmission in a calf. So there the cow was culled, and we found out that the calf was actually positive. There's been no links established between the Norfolk temporary control zone and the Kent control zone. And at present, we've not had any evidence of circulation in the UK midge population. And there's been a very low vector activity in the traps. So disease control, obviously our primary short-term objective was to establish whether there was virus circulating or not in the midge population, hence the amount of surveillance we've been doing and continuing. It always starts with a temporary control zone, which initially has been put in at that 10 kilometres. And then for each of our cases that we find, we do full investigation, epidemiological investigation, and we'll trace all animals connected to that premise. And so what we found till now, the current hypothesis that's been found after all those investigations we've done, was that it's probably been introduced by infected midges that were flown over from the continent, from the Netherlands during September and October. And then obviously we are all aware that the Netherlands, they started this in September, but they've had over 5,900 premises to date with Bluetongue 3, BTV3, and Germany and Belgium have also had cases. So our disease control surveillance continues in both the temporary control zones. We're near enough finished with the Kent one, the extended Kent one, and continuing on the Norfolk one. Our visits include inspections and samples taken from all the susceptible animals, except for lambs under 3 weeks of age. They're exempt. If we do have positive cases, young lambs are also tested on the restricted farms. And then we also do record checks just to see if there's any changes in production that might indicate there was a problem previously. We've taken up till now about over just under 30,000, but probably with today, we're over 30,000. And like I already stated, we continue surveillance. Strangely enough, we still find holdings in the zone that have not been registered. And to tell you the truth, it's often people who have no idea that they should register. Last week I had someone who had horses, and they have a small herd of sheep, and they just didn't know. So just to encourage when you're out and about, just to remind people if they do have sheep or a single cow or something, that they do need to register for this, and there is guidance for that on the gov.uk website. Movement controls hasn't changed a lot. We still have the same licenses. There's a derogation for the dairy cattle. All moves have to be licensed. And within the TCZ, there are moves that to go outside of the TCZ to only to designated abattoirs. Even so, and I think I mentioned that last time as well, for welfare reasons, we do allow a certain amount. We do allow moves out of the zone, but within close to the border of the TCZ. But there is, we do need to know this is a real welfare reason. And with that comes post-movement testing, either pre-movement and post-movement testing, or just post-movement testing, depending on the situation. But the other license conditions also apply. There is a time needed for these licenses, so just if you can encourage, if it's your clients, to apply well in time and at least 5 days, because risk assessments need to be undertaken to be able to understand if the move can be done or not. So types of moves, like I said, from slaughter to designated abattoirs, and then welfare reasons. And if you suspect disease, this is our number. So, and I'm sure you've seen this quite a few times, and hope that, that's it from me for the update for today.
Thanks very much, Sasha, for that. Just while we maybe transfer over to the next presentation for the next speaker, there's a couple of questions that have come in. The first one is really around animal welfare. What happens if you're in an exclusion zone and you have no further grazing, and that is becoming a pressure for feeding and the requirement for grazing? What are the options in these situations?
I suppose, it's difficult. It's trying to find land within the TCZ is the best option for welfare. If there's no feed, try and find additional feed to be brought in. But if that's not possible, then finding land within the TCZ, it would be the best best way forward. If that can't be found, I suppose the next way would be to find land just outside of the TCZ. I mean, welfare, as I'm assuming you all realise, that for us is paramount, and we make sure that those moves can happen. But it does take a bit of, we have to look at risk assessments, and we have to look at the situation to allow that move. So, and also, if the place is outside of the TCZ, we would put that under restrictions as well until samples are taken.
One other quick question that we might just take now as well is why is the TCZ limited to 10 kilometres if we think that the culicoides midges could be dispersed far wider than that? If you could just maybe give a little insight into that as well, please.
I don't know a lot about midges, but in any case, the midge plume would have come across and would have landed at just on the coastal line. And we don't think there is any circulation at present in our own midges. So in essence, it would be there where it fell. And so we were hoping that the 10-kilometre would be enough of size of zone initially to understand where disease was. So hence the reason. But yeah, it's a difficult one, and it was decided that with the disease consultants and the epidemiologists that was the best way to put the 10-kilometre in initially.
Excellent. And it's also maybe worth pointing out that there is wider surveillance ongoing in the south of England, looking more widely for evidence of BTV as well. So it's maybe worth pointing that one out as well. Okay, thanks ever so much,
Yes,
Sasha. Well, maybe. Sorry.
Actually, Colin, I'll add to that we're actually looking beyond the 150 kilometres. We're doing surveillance on that as well. So it's actually quite a lot wider. So yeah, just to note that. So it'll be interesting to see.
Brilliant. Thanks, Sasha. We'll call you back in at the end because we'll have a Q&A at the end. So thanks very much for your presentation. What I'm now going to do is introduce our next two speakers who are going to basically follow on one after the other without me rambling on in the middle. This focus now is moving slightly away from Bluetongue and onto Schmallenberg for very good reasons. One is that there have been reports of increased number of cases in England this season so far. There are obviously links with Bluetongue in terms of it's also a vector-borne disease, and some of the ways it can present clinically in young lambs or calves may look similar as well. So we've got two speakers tonight who are able to give us an insight into both pathogens, both diseases, what to think about on your farm. So our first is Rachael Tarlinton. Rachael is Associate Professor of Veterinary Cellular Microbiology at the University of Nottingham. And she's going to provide an insight into a little bit about the biology of Schmallenberg and what to look out for and how it might impact herds and flocks. And then that's going to be followed immediately after by Vanessa Swinson. Vanessa Swinson is the lead of the cattle surveillance group for APHA and is coming at this from a disease investigation perspective, looking at what to look out for both Schmallenberg and also potentially Bluetongue virus infected cases in very young lambs and calves. So over to you, Rachael, and thank you for your presentation in advance.
That's all right. Hopefully everyone can see that and hear me okay. Someone shout out if there isn't.
That's great. It's working fine.
Yeah, cool. As Colin said, why we're talking about Schmallenberg and a Bluetongue series, Vanessa will also go into this. It is because there's considerable overlap in the two diseases and things that you need to be aware of about them. So what is Schmallenberg virus? It's an insect-borne orthobunya virus, which doesn't mean much if you're not a virologist, but it really only affects ruminants. It's very similar to viruses that have been known about for a long time in Australia and Japan. Akabani virus, I saw my first case as a 6-year-old in 1981. And the really important thing for this seminar is both Bluetongue and Schmallenberg are spread by the same biting midges, by Culicoides species midges, the same species in Europe. So Schmallenberg originally first appeared in Europe in 2011. We don't know exactly where from. However, it's been similar things have been detected in Central and Northern Africa, and we're assuming that's where it originated. There's historical evidence of it in parts of the Mediterranean like Turkey or Israel. Since 2011, it has become established in Northern Europe, and we're now stuck with it. Other important things are it doesn't infect humans, it's not zoonotic, it only causes mild disease in adult animals, unlike Bluetongue, which causes quite severe disease and death in sheep. Schmallenberg doesn't do that, but its biggest effects are reproductive ones, particularly fetal deformities, and that's what we're seeing at the moment. So acute infection in adult animals. In cattle, sometimes it will cause fever, milk drop, and diarrhea. So this little beautiful graph over here is from the University of Nottingham's herd, which is on a robot milk recording system, and we can see quite clearly for this animal when she was infected where there was quite a dramatic milk drop, and you can pick that up on a herd level as well if you're using those kind of monitoring systems. It tends to spread a lot more explosively than BTV. We don't particularly know why. Probably that's to do with the way it replicates in the insects. So it does have an impact, an economic impact in terms of milk drop in dairy herds, but it's quite a short-term disease. They get it, they get over it, and they generally develop quite robust immunity to it. So it's quite an acute disease in adult animals, and generally a mild one. We usually don't see clinical signs in adult sheep at all. What we do see, however, the big effects are in the fetus. So Schmallenberg and Bluetongue, both of these viruses will cross the placenta quite readily, and they tend to replicate in the nervous system of the developing fetus. And this is to understand the pathology. If you understand that, it makes sense. This tends to happen in the middle trimester of pregnancy when the brain and the nervous system are developing, but before the fetus is able to mount its own immune response. So the virus just replicates away in the nervous system and basically just damages it so it doesn't develop properly. So this picture we've got here is from our pathology service, and these are lambs, I think they're all lambs, yes, that were born live, born affected by Schmallenberg. So the ones that were close to birth when they were infected up the top look relatively normal with their brains. The ones that are infected earlier in the brain development have virtually no brain tissue at all, just big fluid-filled sacs. So that's called hydroencephaly, when they have very little brain tissue developed and just big fluid-filled sacs. Because the brain doesn't develop, it doesn't stimulate muscle movement. So that fetal movement's really important for muscle development. So the muscles don't develop, so the farmers will report that as very skinny, long alien-looking legs, because the muscles are not moving, the joints don't develop properly, and they are fused. They don't develop, and you have those fused fetuses with fused legs, fused spines, very twisted, fused fetuses. The ones infected very earliest on in pregnancy might be aborted, and the first sign that something might be going wrong is generally an abortion storm. Fetuses that are infected very late in pregnancy in the last trimester are able to mount their own immune response to the virus, so they may clear it, they may get rid of it, they will generally be less affected by it. And those animals are more likely to be born live. So the worst deformities are seen in sheep that are infected between about day 28 and 40 of pregnancy, and in cattle infected from about month 3 to 6. That's very similar to what happens with Akabani. Where are we up to? So Vanessa will go into this in more detail, but the students have sent us various gory pictures of the types of deformities you will see. So dead fetuses, aborted fetuses, fetuses with fused skeletons. These are the technical words for these terms. Fetuses whose brains haven't developed, their heads may be deformed. They may be born live, but unable to suckle, unable to stand, or blind. Importantly, for welfare reasons, because those animals just don't have the brain tissue, they're unlikely to get better or to be able to learn how to do these things. So they're mostly going to require euthanasia. Nursing them or attempting to nurse them is often very, very frustrating and unrewarding. Undershot jaws, brachynathia is seen a lot. Because of particularly the very fused fetuses, giving birth is often quite difficult. Dams may require assistance, there may be significant birth trauma, so cutting the fetuses up or a caesarean may be necessary during the birthing process, and that's another impact. The other thing to remember is not all deformities are Schmallenberg. There are things it doesn't cause, so it won't cause extra limbs, it won't cause gastrointestinal problems or skin or heart defects. So not every deformity is Schmallenberg. It's this quite typical pattern. You can get only one fetus in a multiple birth infected, with the other fetuses born normal. And I'm not going to go into this tonight, but if you're really interested, there's a YouTube video from the 2011 initial outbreak from the vet school in Hanover, which has some really good video footage of live-born dummy animals, what they look like, what they behave like. It is in German, and the footage is about halfway long, but it's probably the best video footage of those cases we've got. So Schmallenberg and BTV have other effects as well. The primary big effect for impact is on fertility. So all of these viruses and BVDV or border disease, for the same reasons, all cause the same deformities. All three of those viruses can cross the placenta and can replicate in the nervous tissue. Because of that, at the same stage of pregnancy, you see the same pathology. The fetuses will look the same damage. You can't work out which one of these it was without doing lab testing, PCR, and or serology testing of the fetus and dam, and that's reasonably complicated because of whether it's best to contact the lab as to what to send in, because you'll need a combination of those from both the fetus and the dam to work out what's going on and which of these viruses is the problem. All three of those viruses also specifically knock down interferon, which is the major antivirus signaling system that the body uses. In ruminants, peculiarly, the maternal recognition of pregnancy signal is an interferon tau. All these viruses knock that down as well. What this means is the animals will lose any early pregnancies. That pregnancy will fail to establish because the dam won't recognise she's pregnant. That will show up as a lot of or an increased number of animals failing to conceive or returning to service for dairy cattle. That's really well established for BVDV and BTV. We think that also goes on with Schmallenberg, but it's been a bit harder to get the numbers pinned down on that. This early fertility problem tends to be much more noticeable in dairy cattle because we monitor their individual cycles quite closely. So female fertility loss, early fertility problems are a big issue. This also affects males and male fertility. So one of the really important trade impacts is that Schmallenberg and BTV can end up in the testicles, which is an immune privileged site. So the rest of the animal might clear the virus, but it can be sitting in their testicles, happily just sitting there. What that means is that they can excrete these viruses for, we don't know for how long, for BVDV that can be lifelong. Schmallenberg, it's been recorded for up to 3 months. This also happens for Bluetongue. It's not been reported in rams or bucks. However, we have tested a much smaller number of animals, so it is still possible. We haven't tested enough animals to rule that out. And what that's meant is that countries that don't have that, or when you're exporting semen, are going to require testing of that semen and or the donor animal, the combination of PCR testing and or serology. What they require is going to depend on the country. We don't have any control over that. It will also affect male fertility, affect sperm quality, affect sperm count. Once again, we know a lot about this for BVDV and Bluetongue, but not so much for Schmallenberg. So the epidemiology of Schmallenberg is quite complex because we have both an annual and a multi-annual cycle. So it circulates in midges. So the timing of when animals become infected depends on the numbers of midges. That depends on the weather. So midges breed, their numbers will build up over summer. Their numbers will be higher in wetter years and higher when the temperature is higher. So it's dependent both on moisture and on temperature and on the time of year. What we tend to, and how windy it is as well. So what we tend to see in the UK is that midge numbers typically peak in or around September. So they'll build up over the summer and we see the maximum numbers in September. Though as we warm up with climate change, that's shifting later and later, and the period of time when the midges are around is shifting later and later. And this tends to be when the maximum risk, when the maximum for these viruses go through, when the viruses spread explosively. So what this means is that flocks or herds that are mating in summer or early autumn will be the worst affected. These animals will be in the susceptible period of pregnancy when the midge numbers are highest. So this graph over here is from our 2017 survey, and it shows flocks that in 2017, which is the last big outbreak we had, which thought or were confirmed to have Schmallenberg. And you can see that flocks that were mating in July and August were the most heavily affected. Those that were mating in September, October were still affected, not quite as many. Those that started mating in November, December, by which time the midge numbers had dropped because the temperature drops, were much less affected. So the timing of mating affects when your flocks are infected. The picture up the top is from 2013, and it's just there to demonstrate that the virus was present across most of the UK and the island of Ireland. We didn't have any data for Scotland, but there have been cases reported in Scotland. The other important annual epidemiological cycle is because of the length of pregnancy, sheep cases will be seen about 3 months earlier than cattle cases. So we expect cattle cases to pick up later in the season. So that's annual cycle. We also have imposed on top of that a multi-year cycle, and this is quite typical of these viruses. And in Australia and Japan, they see multi-year cycles of this. What happens in endemic areas in Northern Australia, which this has been best studied, these viruses circulate annually. Most animals will be exposed before they ever become pregnant in their first year of life, and you only see the occasional sporadic cases because that herd level serology stays high because it circulates annually and you don't have huge numbers. That's different in southern Australia where the midges are not in high numbers annually and it doesn't circulate annually, and they see what we're seeing where at random year intervals you get really explosive outbreaks. So the midges have spread and there's lots of animals that have never seen it before. What we appear to have in the UK with Schmallenberg is the worst case scenario, where we're on the edge of the Schmallenberg endemic zone. It's a little bit difficult to tell because no one's done consistent monitoring, but the serology figures for annual circulation in Europe are much higher. So probably on the continent, they have a more stable situation, a more stable endemic situation where it circulates much more often. Animals mostly get it early on and they don't see the number of cases we do. Because of our weather, climate, where we are, we're not seeing that. We're kind of on the edge, and that means we're susceptible to big explosive outbreaks. And this is what's happening. So went through in 2012, and Vanessa will present this figure later. We had no circulation, so nearly everything seroconverted. It disappeared by 2014-15. We saw almost nothing, so 70% immune. Then as these animals were culled out and their immunity also dropped with time, with animals that weren't culled, we got to a point where we had virtually no immune animals about 4 or 5 years later. And then we had a new outbreak, blew over from the continent, and it took off again. And the same thing happened again, it dropped off. Small uptick in cases in 2021, 2022, and now we appear to be in a situation where we're back to where we started and we've had a new outbreak. So probably we're going to be looking at this, and so far this is panning out about every 3 to 5 years. How big, how often will depend on that combination of weather, midge abundance, and the number of susceptible animals we have. And what the bottom graph shows is these were animals that were part of something else altogether, where we happened to be collecting serology samples for them in 2016 when this went through the last time in a large outbreak. And the blue ones are used that had been alive during the first outbreak, and you can see they developed a really big, massive jump in their in their antibody response, in essence, they got a new booster from a new infection and had a massive antibody response. Ewes that were born later had no pre-existing immunity and didn't develop as good a response. So that's hopefully helping to explain why we see this really unstable multi-year cycle imposed on the annual one. So impact on farms, this comes from a survey we ran in 2017, and just a plug, we're going to be rerunning this survey, which was sent out to farms to look at what the impact was on their lambing in 2017. So we had quite a lot of farms responded in 2017. About half the farms that responded, a bit under half, thought they'd had it. And of those that thought they'd had it, maybe about 30% of them had actually had testing done, and only about half of those were actually confirmed on the testing. There could be a lot of reasons why you might get a negative test. It doesn't mean they weren't affected. So the biggest effects we saw were on lamb mortality and, to a lesser extent, the number of ewes requiring assistance. It had quite an economic impact. It had a much bigger emotional impact. The deformities are quite spectacularly horrible to see, and that's something that shouldn't be underestimated, is that this has a very big impact on the people dealing with that. In terms of economics, so the lamb mortality, that's what this set of graphs here was, on average 3.5% higher in flocks that were affected than flocks that weren't or didn't think they were. But that kind of hides a very wide spread of impact. So the bottom graph is the flocks that didn't think they had it, and that's kind of what sort of lambing impact losses you'd expect in a normal year, and there is a bit of a spread. What you can see with the flocks that thought they had Schmallenberg is a much bigger spread, with some flocks affected very, very badly, and some of that will be in flocks that have synchronized mating that happen to be hit at exactly the wrong time. So the impact can be very, very high for some flocks. So the average impact kind of hides a lot of variation in the figures. But it really can be quite devastating for flocks that are heavily impacted. Other ruminants, we do see fetal deformities in goats, the same as in sheep. Goat herds seem to have a lower seroconversion rate. The midges, by and large, don't seem to like goats as much. We do also see seroconversion in pretty much all the ruminant species we've tested, wild and zoo, including the various deer species we have in the UK. No one's ever reported a fetal deformity in wild or exotic ruminants. That doesn't mean it doesn't occur. We don't usually find aborted or dead fetuses from wild ruminants because they get eaten by foxes. There is a big question as to whether deer form a potential vector for it to keep circulating, regardless of what's happening in the cattle and sheep population, and that question's still quite out there. We see lower seroconversion in deer than in cattle or sheep, but who knows, because we don't have consistent monitoring of that. Control. So vaccination, when it was available, was very effective at preventing fetal deformities. There were 3 killed inactivated vaccines originally released, but because of that multi-annual cycle, the uptake of these was never high. Animals that have had a problem in 1 year will likely be immune and will not likely have a problem the next year. Because of that, the vaccine uptake was really never very high, and the companies realistically were making a loss on it, so stopped producing it. So there have been none available since 2016. None are currently in production in Europe. So there were a number of other reasons why the vaccine uptake was problematic. It was a killed inactivated vaccine, required 2 shots. It wasn't licensed for pregnant animals, probably perfectly safe, it just hadn't been tested. The cost was too high to be economic for most sheep flocks, And the big factor was lots of uncertainty about whether it was necessary, whether it was going to come back or not, whether it would disappear. Blocks that had been heavily affected or had seen neighbours who were more likely to vaccinate, but by and large most people didn't. In the absence of a vaccine, the only other realistic option for reducing the impact of this is moving the timing of mating until later. There's been quite a lot of work looking at insecticides on midges, that kind of thing. It doesn't really it's very difficult to do. It doesn't really have a large enough effect to make a difference. You're talking about field scale, and it's just really, really difficult to do, plus the environmental impacts. So really the only realistic option is looking at your timing of mating. So this is pretty much it. So this is where we're at for this year. Vanessa will go into what we have so far. It's very early in lambing, but we do appear to be having quite a large outbreak this year. Based on what's happened in the previous outbreaks, the early mating lambing flocks will likely be worse affected. We will start seeing cattle cases probably in about March onwards. Previously, we have seen fewer cattle cases than sheep. One of the things that may be different this year is it didn't frost till very, very late. The midge numbers were very high until very, very late. So that may be a more drawn out period than we've seen before with higher impact. If it behaves like it has previously, we're likely to see very little circulation of Schmallenberg next year. And the animals that have had problems or were exposed this year are unlikely to have future problems. The important thing in terms of BTV consideration is if a farm was Schmallenberg affected or near to one, because they're spread by the same midges, they are likely to be vulnerable to BTV3. And the two viruses, there's no cross-immunity, cross-reactivity. So immunity to Schmallenberg has nothing to do with BTV. But because it's the same midges, you're likely to be susceptible. And geographically, its cases are rarer in Scotland, but we have seen them. There is nowhere in the UK that we could say is not going to be BTV affected. It does tend to spread over a longer time frame and not as explosively, but it's very hard to predict because it's very weather dependent and very dependent on midge numbers. The other thing is testing is really important because you can't tell the difference in the fetal deformities between these three viruses. This year, given the timings of things, these fetuses this year will not be BTV because it wasn't circulating as far as we're aware, and we're pretty confident of that. We would have seen that at the time that SPV was going around. Next year, that will not be the case. And BVDV and border disease, which are both endemic here, will cause the same problem. So that testing is really important. So that is it from me, and this is just an acknowledgement slide because there's been we've been working on this since 2011, since the original outbreak, and there's been an awful lot of people worked on this over the years. So that's it from me. So I'm going to hand over to Vanessa now, who will, who will be going through where we're at with the current Schmallenberg outbreak and giving you where we're up to with that, and then we'll have some questions afterwards. It's all yours, Vanessa. You are on mute. You're still on mute. She's off now. Right, good. We can see that. All good.
Okay, hopefully you can see me and the presentations in presentation.
All
Perfect,
good.
Vanessa. Thank you. Yeah.
Thank you. Yes, sorry, I was having problems unmuting myself, but hopefully it's all right now. Okay, so I'll hopefully not duplicate too much of what Rachel said, but there might be a little bit of overlap in our presentations. So as we've seen from the graphs, there are these peaks and troughs of Schmallenberg that we've seen since 2012. We saw a few cases in 2021 and 2022, so we weren't really sure if that was kind of it for this 3 to 5 year cycle, and whether there was quite good immunity out in herd and flocks, and then we wouldn't see much this time. But we're currently seeing evidence that there was quite a lot of Schmallenberg circulating in Great Britain in late summer and autumn of 2023. And as Rachel says, one of the reasons for talking about this tonight is that the fetal deformities detected from these two and other diseases like BVD and border disease are very similar. As we've also heard, they're both spread by the same midges. We've heard about reports of sheep flocks recently experiencing significant losses from Schmallenberg. As Rachel's just told us, the challenges for prevention and control are very similar. And we're likely to see calf cases in spring 2024, 3 to 5 months from what we're seeing now. And it's important to investigate these for a variety of reasons, including checking for Bluetongue. So just a bit of recent history from what we've been seeing in the surveillance network. So in November 23, there were reports from 3 labs, including APHA, that there was high milk antibody levels. Some herds that had been either negative, seronegative in the milk, or had low levels, then suddenly had increasing antibodies to Schmallenberg in both England and Wales. From beginning of December till the middle of January, we had 63 cases where submissions were sent into our lab network of labs, and these have been post-mortem and checked for Schmallenberg. The testing results we've got so far, 33 out of 41 were PCR positive for SBV. All of these were malformed and stillborn, and the majority that we've seen so far were lambs. I've put there some of the counties that we've recorded cases in, but that's not an exhaustive list. There have been other counties affected since then. So just a little bit more on the recent history as well. So as you've seen, we've had reports of this or submissions for the last 3 years, but at quite a low level until now. We do, as part of our surveillance testing, look at dam serum samples, particularly if the PCR is negative, but the pathology is suggestive of SBV. And you can see the percentages of samples that were positive on the right-hand side there. We've had submissions to look at the deformities, which I'll show you some of the photos of in a minute. And as Rachel said, in our VIDA diagnosis, we've seen these cycles of every 4 to 5 years. And there's different reasons, or again, as Rachel's discussed, about why you might be seeing this spike in cases this year. So, sorry to anybody that's having their evening meal. I'm afraid we've got a few gruesome pictures coming up. So these are the range of things we would see in small ruminants and camelids, potentially where Schmallenberg is involved. So early embryonic loss, we've had reports of poor scanning in some flocks. Abortion, which the things in red there indicate where there's a whole variety of things that might have caused abortion and early embryonic loss, but they would include Schmallenberg, Bluetongue and border disease, and we would include those in our differential diagnosis when we're investigating these cases. They're more sort of the topic for this evening as far as pathology is fetal deformities with abortion, or stillborn lambs with deformities, or again, as we've mentioned for Bluetongue as well by Peter Mertens, we can get these dummy lambs born which have CNS damage but are born alive. And same for cattle, so the small font there is the ones I've just mentioned, which would be the same for cattle. In addition, as Rachel's mentioned, we can see, particularly in dairy cows, pyrexia, mild malaise and milk drop and diarrhea in cases of acute Schmallenberg infection. So just going through some of the different deformities that we might see on farm or in the PM room. So first, the arthrogryposis or bent and fixed limbs. These are some typical cases. Some of these are from 2012 or 2017, and some are ones we've seen in the last few weeks. The next ones here are spinal deformities, torticollis kyphosis. So there's some images over the right hand side there showing the spinal deformities. Also mentioned by Rachel is the jaw deformities, usually the sort of overshot or sort of parrot mouth type of appearance. And then one of the main reasons for discussing this tonight is the brain deformities can be quite similar between Schmallenberg and Bluetongue and the pestiviruses like BVD and border disease. So sort of working from the top there, we've got, you can see the cerebral hemispheres have a collapsed appearance. The top one, top right, we've got cerebellar hyperplasia and reduction in the diameter of the spinal cord as well. Bottom right, we've got sort of fluid-filled, thin-walled cerebral tissue there. Again, in the middle at the bottom, collapse of the cerebral hemispheres. And then the view you can see there of the spinal cord, where it's sort of been cut at the epiplantar-occipital joint. You can see that the spinal cord is quite a lot smaller than in diameter than it should be relative to the spinal canal. So that's quite a good way of assessing the spinal cord and sort of what it looks like relative to the structures around it. So now just sort of moving on to differential diagnosis, Schmallenberg, and why we're sort of trying to advise everybody to get these investigated and not assume it is Schmallenberg. I think if they've got small ruminants have arthrogryposis, then Schmallenberg is the most likely diagnosis, but that's not to say all of these will be that. So again, got to consider Bluetongue at the moment, and certainly going through into 2024 and 2025. Border disease, which also can have significant effects on the flock if that's going through. Deficiency such as copper maybe would have some gross pathology in a few cases. Some of that might be only more evident on histopathology, the effects of low copper in pregnancy. Toxins and systemic sort of hypothermia in the dam, toxoplasmosis, and musculoskeletal deformities and genetic conditions. The bottom right there is twin goat fetuses that were submitted to Thirsk, and you can see in the right hand side kid brain there that there is hydranencephaly, and that was sort of checked for Bluetongue, but the diagnosis was toxoplasmosis, and the dam was low in copper as well. So moving on to cattle, of which there sort of seems to be a wider variety of conditions that could look like Schmallenberg, particularly with BTV8, and some of these are borrowed images from other people, but you can see there there's a real lack of cerebral tissue and just a sort of fluid-filled sac there in the blue image, and then top right, virtually no brain tissue at all from Bluetongue. All of the bottom images are different cases that we've had through the labs over the last 3 or 4 years, which were BVD related. So at the left there, the cerebral tissue is much reduced, collapsed in the 2 images in the middle, and then the right there you can see that the cerebellum is very much smaller than it should be. Some other things that we've seen over recent years, we've seen a few cases of Arnold-Chiari malformations. It is seen in other species, including humans. Not quite certain what causes it, whether there's genetic element to it or maternal nutrition, but that can cause similar limb deformities, and also brain abnormalities as well. The middle 2 images there, Schmallenberg was ruled out on those, and the diagnosis was likely related to plant toxins, teratogens during gestation. And then on the right there, we've got 1 or 2 genetic musculoskeletal and CNS deformities. Another one that the cattle group's been dealing with recently is poroncephaly in Limousin. So you can see there, top left, this collapse of the cerebrum is very similar to what you might see with Bluetongue or Schmallenberg or other diseases as well. And that's in the middle there is a fixed brain with one of the Limousin fetuses or stillborn Limousins where there's lots of fluid-filled cavities in the brain. The right there, that was more of a musculoskeletal one, which was submitted as a possible Schmallenberg and was a genetic osteogenesis imperfecta in stabilised cattle. So just moving on just briefly to investigating these. So I sort of put the different options there, which so will be dependent on sort of how likely it is maybe to be Schmallenberg, where the farm is located, if it's possible to get full fetus and placenta into a investigation centre. So certainly, you know, for vets doing on-farm examinations, if you're sort of just mainly looking for Schmallenberg, you could do placental fluid scrape, collect some fetal fluid from a body cavity, have a look at the brain, which is probably easier in lamb brains than it is for calves. So sort of we'll just move on to maybe taking the skull off in a minute, but that can be, you know, in a stillborn or neonatal calf, less easy if you haven't got a vice to put the head in and to saw off the top of the skull, and then taking a section of hindbrain to store for the PCR test. Then sort of on farm, you know, if it's sort of less likely that necessarily is Schmallenberg, but that is one of the differentials, then you would be needing, if you're going to do an on-farm PM and taking samples, to be getting the full range of abortion investigation samples. Sorry, I should have said at the beginning, it's, you know, I think all of these cases, it's great to discuss them really with your local centre or your local lab, because, you know, we're always happy to talk these cases through and advise on sampling and discuss, you know, which samples are suitable, whether it's more advisable to send in the whole fetus and placenta rather than just samples. So then, you know, we've got obviously the post-mortem provider, which is on the left there, as an example of one ready to sample. But in addition to that, it's worth considering maybe at the time you're on farm looking into these, to taking some bloods from some dams in case we need to do a bit of serology testing as well to help investigate the issue. And then just excuse me, just going on to sort of other investigations, so lambs and kids that are born alive with deformities, or sort of dummy calves or lambs, often it's sort of freshly euthanized, but also by injection rather than to shooting, so that the brain tissue is intact, submission to a PME provider, or if it's on farm, some blood before the animal's euthanized, or otherwise serosal fluid, clinical exam, sort of look at what deformities are sort of evident in the live animal in terms of neurological deficits and things like that. And then post-mortem, including examining the brain, maybe looking at the atlanto-occipital area, at what the spinal cord looks like. Then sampling brain, spleen in case you want to check for things like BVD or border disease, and then trying to possibly fix the brain or any other tissues that are of interest in case we need to do histopathology later in the investigation. As far as adults and post-weaned, if we've got any sort of signs suggestive of Schmallenberg, it's definitely one of those where we sort of need to look at all the different differential diagnoses, really, because we need to get the sort of complete picture for the main differentials. So looking at things like salmonella or winter dysentery, depending on sort of time of year and risk factors. And then if there's any acutely affected animals, bloods could be taken, EDTA or serum for the PCR, otherwise paired serology. As I've just mentioned, with the fetal deformity, sometimes people do submit bloods from dams as well, and we are offering some free of charge testing for PCR testing on the brain tissue fetuses, but we're also offering some antibody testing for dams. In some of those cases, we wouldn't have the paired serology, the acute and convalescent, so it would really, and also because the fetal deformities are a historical event, we wouldn't have any that are likely to be acute at that time. So the dam serologist helps once other causes have been ruled out, and if it sort of either can help to rule Schmallenberg in or out, or make it more or less likely, depending on sort of the antibody, if antibodies are present, and what the level of antibodies are. And just, so I borrowed a couple of Rachel's images here, you'll recognise them. So just remembering about breeding animals and potentially the effect on fertility, and APHA are involved in a lot of PCR testing of semen for Schmallenberg, because it's it's quite a big thing now for export of semen, as to whether what surveillance that bull has had for Schmallenberg, and whether the semen has been PCR tested for Schmallenberg as well. So in summary, sort of from Rachel and myself, so current evidence is that there's going to be a peak of incidence for this current sort of lambing season and spring calving, and then maybe a little bit more later on as well in a year's time. It's important to investigate these and not assume they're Schmallenberg, because quite a few cases that we've had through, show that's not necessarily the case. And we want to keep an eye out for Bluetongue 3, but also we've got to consider that BCVA and AHDB, epizootic hemorrhagic disease, are both in France, so we might be looking at risks from around those, unfortunately, over the next year or two. Increasing monitoring of Schmallenberg and sort of checking maybe bulk milks or bloods sort of in advance can help management decisions, and see whether the time of mating could be altered on individual farms, but also sort of monitoring the situation with Schmallenberg can help to give overall industry advice and look at how much vaccine uptake there might be in the future, if that seems to be the only way of controlling it. And on the cattle group, I know Colin will agree with me on this, that we always say that ruling diseases out in these sort of investigations of abortion and fetal deformities is very important, as well as ruling diseases in. So I think that's Just maybe an acknowledgement slide there. So yes, thank you to all my APHA and Cattle Group colleagues for their help and sending me lots of good images. Thank you.
Thanks very much, Vanessa and Rachel, for what have been very detailed and very informative talks on Schmallenberg. Perhaps if I could just ask, well, Rachel, put her camera back on, so we've got everybody back on screen. And we have got a list of questions that have come in throughout the talks, and maybe in the next 10 minutes we'll try and get through as many of those as we can. Also, don't worry, we've got a register of all the questions that have been posed, so if we don't specifically on the webinar tonight, we'll try and answer it offline. So first and foremost, and I'm going to address this one probably to both Vanessa and to Sasha, and you can choose which one answers it first, both from a farmer perspective and also from a vet perspective. We're now pulling this back to the Bluetongue story and the possibility of identifying Bluetongue virus infection in a deformed calf or lamb. So if you're seeing lambs born with obvious deformities or neurological signs, or calves born with those deformities, firstly, from a farmer point of view, what should you do? Who should you notify, or should you do anything? And then also, if you could answer that from a vet point of view, if you're looking at it from a sort of diagnostic perspective.
Well, I would say in the first instance to talk to your private vet, and then encourage them to phone up a lab or a local PM provider to discuss the case and decide, whether sampling is appropriate. I think, we wouldn't in these cases be automatic. Might be Bluetongue. We would, there would be, discussions about Schmallenberg and testing for that, or and checking the brains of affected fetuses before there wouldn't be any other sort of notifiable disease type of action until it had been looked into, unless of course there were any clinical signs in adult animals that were suggestive of anything of concern.
Yeah, I agree with Vanessa. I mean, obviously that they're not of any notifiable disease I can think of, but pyrexia, obviously in cattle, which can happen with Schmallenberg, would be one for Bluetongue. So yeah, so be looking for symptoms that would be indicative of notifiable diseases. But you can also discuss that with the VI centres, because they would really help whether it would be notifiable or not.
Thanks very much. So that's quite clear.
Colin,
go on, Rachel. Yeah, please.
Right now, they're not going to be Bluetongue. Right now, they're going to be Schmallenberg. That will change later in the season when both are possible. You are very unlikely to see Bluetongue without clinical signs in adult animals because it is such a severe disease. So you would know if you'd had Bluetongue through your herd. It's just that fetal deformity thing later, which will be later on, and this time next year we'll really be looking at that.
Great, that's very useful. So we've got quite a clear steer that if you're seeing these deformities as a farmer, as a vet, is probably first port of call would be to do some further disease investigation, looking for the full range of differential diagnoses, and then if BTV couldn't be ruled out, then that might be considered at that point, but also a steer that it's not likely to be a significant issue right now. Okay, that's really, really useful. Another question relating, I think, really to clinical signs. There's a large focus on clinical signs associated with poor fertility and dystocia. How often are these clinical signs the trigger to disclose BTV cases? Perhaps Rachel, do you want to come in there?
Those signs by themselves wouldn't trigger a BTV notification. An acute BTV case, you will have their cattle and sheep. That's much more likely to trigger an investigation. So the fertility issues by themselves would not be making you think BTV unless you've got all the other things that go with BTV, really high fever, excoriation, swollen heads, sheep. So that's where you'd be thinking BTV rather than another cause of abortion.
Excellent. Thanks very much. Question for Sasha, I think, and it's related to clinical science. Have any of the cases of positive BTV so far shown any symptoms?
No, and I actually thought about that after my presentation. I should have added that in. We haven't had any clinical signs at all, which is, but then if you think about it, in Holland, also a lot of the animals were not showing any clinical symptoms, yet they've tested positive, so for serology, and no one's ever noticed anything. So yeah, it's possible, and we're at the minute, I said 30,000 samples, and we've had just over 50 cases. So it shows how small that is in comparison.
Excellent. Thank you very much. Another just question to Rachel. Can theoretically midges carry both viruses? Be a very unfortunate midge to carry both Schmallenberg and Bluetongue. Is it possible?
Yes, yes would be the answer. Do we have an answer of whether we've actually detected it? No, because we haven't had a situation where we've had both circulating and testing. But yes.
Thank you. It'll be quite some midge, I guess, but thank you for that. We've had a few questions on, I suppose, arising from Sasha's talk about the zones and the regions and animals moving in and out of zones, probably following on from the questions about welfare. Maybe just rather than to answer all of the specific questions that we've had in, could you make any comment about some of the decision-making processes that you go through with considering licenses, both in terms of risk of animals moving in or out of the zone, and also in relation to need and welfare and all the rest of it? What's the sort of process that the licensing team are going through?
So the licensing team look as to where the animals are coming from and where they're going to, so the areas that they've come from, and in relation to what is in and around them. So it's a veterinary risk assessment that's undertaken. So it's it's looking at risk movement. So whether it's high risk to low risk to high risk, and any risk that would make it not a good decision to move the animals. There's also, when do we release these? It's also looking at identification of animals. So there's a lot that goes into these thoughts, also with policy, whether we can allow these moves. Legislation is very clear with TCZs. We're not allowed any moves without licenses, and we're trying to contain the disease as much as we can, and try and find any we have, so that if and when we start again in the springtime with more midges, that there'll be less that there won't be any animals infected out there that can infect our midges to spread the disease. So there is this need on control on shifting animals to try and minimize the risk for the rest of the country as well.
Thank you very much. And I'm going to stick with you, Sasha, if I may. Another, I suppose, linked question. What is the likelihood that we will go down the route of zoning or regionalisation so that we can declare some parts of the UK BTV free? It's a big question to perhaps ask at this stage of the year, but have you got any comment on that just now as to sort of perhaps longer term thinking?
We are currently looking at it, and discussions are ongoing. There's been a lot of discussions as well with trade partners, because that is also an aspect to this, and also in disease risk, and how far do we want to, what is the necessity, and what is the risk to the rest of the animals. So it's also understanding the pressures of zones themselves, and how that works. So there are no quick answers to this currently, but we are working on it, and hopefully within the next few weeks we will have further answers.
Thank you very much. Question for Vanessa, please. And it links to the European situation. Have there been cattle or sheep that you're aware of in Europe that have been found BTV3 positive that have shown any specific fetal abnormalities? So following on from the sort of things you've been describing in your talk.
We did go to a webinar a few days ago with some epidemiologists from the Netherlands. They haven't seen at the moment many fetal deformities of BTV3. They also haven't had that much Schmallenberg so far either. It's still quite early for their lambing season over there, so they may well see some soon, and certainly in the cattle as well, going forward into the next few months. We don't really, I think we're not as sure about the fetal deformities from BTV3 as we are from other Bluetongues like BTV8, and also not was, with our breeds or our climate, whether that alters at all what you might see. But I don't know, Rachel, what's going on? Not at all, but so unfortunately, no. The short answer is we haven't got much information to go on from the Netherlands. We've got a lot of information from the Netherlands on a lot of the clinical signs and morbidity and mortality that they've seen over there in cattle and sheep, but not that much on the fetal deformities as yet, I'm afraid.
Okay, thanks, Vanessa. Rachel, have you got anything else to add on that? Or I know it's very early days with all of this because we're still learning about BTV3 compared to other Bluetongue serotypes. So anything more to add from your perspective on that? Oh, Rachel's gone. I'll maybe, if she pops back, we'll ask her that question. Just maybe also to say the intention for a future webinar, either the one in 2 weeks' time or perhaps 4 weeks' time, is to bring back some colleagues from the Netherlands to talk a little bit more about what they've been seeing, both in the early stages of the outbreak and also, what they're seeing now in relation to fetal deformities. So we'll hopefully, through one of these webinars, be able to give a bit more of an update from what's happening in Northern Europe. Rachel, you're back, which is great. We sent you packing with the last question. Do you have any more comment on fetal deformities seen in Northern Europe? Just picking up on what hopefully you heard Vanessa say.
She appears to be having audio issues, Colin. I'm trying to troubleshoot in the background, so carry on for now.
No problem. Right, we will carry on for now. Question then for either Vanessa or for Sasha, and this is, again, it's a forward look, I suppose, trying to look at the potential course of BTV later this summer. Do we have any information on surveillance in the Netherlands prior to the outbreak starting in September? Were they seeing any initial warning signs in the run-up to their first clinical cases? Is there anything that would give us the early warning here? Sasha, do you want
Shall I let you go, Vanessa? Because we heard the same webinar, so.
Yes. So Sasha can fill in on the details that I've forgotten, but certainly they have looked back on bulk milk samples, and they don't think it came into the country much before sort of end of August, early September. They also did see retrospective. Now they look back at milk production. They have seen milk drop around the time different areas of the Netherlands got affected, which was quite, looks quite significant when you look at the maps and how things changed with time. Sasha, I'll hand over to you.
Yeah, no, I think that's what they said. I think they said the indication was about, or early warning signs that obviously they didn't know about because there were not a lot of clinical signs, was about, they stated several weeks, so maybe 2, at most 3 weeks beforehand. So it really hit them. And so it wasn't there for a long time before they had all the clinical signs and problems.
Thanks very much. And it's certainly something that I think we're going to be very aware of as we run through the summer as possible early warning signs and how to perhaps investigate them. I'm conscious of time. According to my computer, at least, it's 22 minutes past 7. So I'm going to go to a final question, which might be a really tricky one to answer, but I don't know whether any of our speakers have any comment. Is there any news from the EU on the availability of a vaccine for BTV3 yet? Is there anything that anybody might want to add on that?
As I understood it, Holland is working very hard on it. They've been looking into it, and as far as I've understood, they're hoping something will be available later this year. But I suppose it depends on experiments and all the testing they have to do. Whether that's next year, who knows? But they're working hard on it in the background.
Yeah. Thank you for that. We appreciate that it's a very difficult question to answer at the moment, and it's clearly what we are probably all looking for. And rest assured, it's a question that will be at the forefront of all of our minds in terms of future control of this virus and passing on information as soon as we get it as to where that might be and what the implications of a potential vaccine might be. Okay, there are a few questions that we've not got to, and I can try and reassure you that we'll try and answer those offline. And thank you very much for a big list of questions, which I've tried to summarize, and your interest in our presentations this evening, which we hope you found useful. So I would like to end firstly by thanking all of our speakers tonight for their time and their effort in putting their presentations together and sharing their knowledge. It's really appreciated, both by the vets and farmers on the line. I'd like to thank AHDB for again hosting this and therefore their patience and perseverance with the likes of myself trying to work these systems. I would definitely like to flag that there will be another webinar in 2 weeks' time, and we will get the details of that out to you and who the speakers are and what we're going to cover. And within that, there will certainly be a further update on any current developments with the current disease situation. And a final plug, if you are a webinar guru tonight, you'll have missed the start of it, but there is currently a National Sheep Association webinar on at the minute on what makes a successful lambing. You can join the second half of that if you want to flip over to a different channel right now, and I think that will also be available as well after the event, so just to signpost that one as well. So thank you very much to everybody for their attendance, and enjoy the rest of your evening. Take care now.